I would like to thank Nevin and Trey for inviting me. I'm really excited to talk about the research that we do in our lab. And we're interested in virus-host arms races, where the virus hijacks cellular genes to convert the cell into promoting replication and spread. And at the same time, the virus has to evade the host defense genes. And on this interface that is often shaped by evolution, a lot of interesting biology occurs. It can also be a target for antiviral therapy. Why are we interested? The last years has made clear that there's a continuous threat to global health from emerging and re-emerging viruses. Ebola, dengue, and Zika really affect global health on a large scale. Viruses are not only bad, viruses are also engineered for novel gene therapy applications, and viruses express their gene products that over evolution have been specialized in manipulating the host. So studying viruses is also interesting in understanding cellular functions. Our lab takes a genetic approach. In host pathogen interactions, usually people start with one favorite host gene and they study that in detail. We're interested in studying all host genes at the same time by doing genetic screens. And for that, we use our do-it-yourself screening toolbox. So we have developed a haploid genetic system where in haploid human cells you can use gene trap mutagenesis to create large-scale human knockout cell collections, and we did that before CRISPR was around, and now we're also using CRISPR genetics to achieve the same thing, make large human knockout cell collections and study them in the context of virus-host interactions. And basically you make knockouts, you expose them to a lithic virus, and you see which rare mutants of the cells survive. And the first problem that we tackled was Ebola virus. In 2014-2016, there was the largest outbreak on record with more than 25,000 cases and an over 30% fatality rate. We don't have a BSL-4 lab, so what we used was a trick. We used the Ebola virus glycoprotein, pseudotyped in a harmless virus, VSV. And this virus enters the exact same way as Ebola virus enters the cell, so this is an excellent system to study Ebola virus entry. This virus has also now been used as a live vaccine in that outbreak. So we have our haploid cells, we mutagenize these cells and create large scale of knockout mutations. We expose it to the virus, we allow the rare mutants to grow out, and we map the genes that are mutated in this cell and compare them to the starting population. And when you do that and you calculate a significance of enrichment, we found a very nicely defined set of genes, around 20 genes. And the top hit of this gene was very unexpected, is a cholesterol transporter, MPC1. So mutations in MPC1 cause a lysosomal storage disease. And the cholesterol cannot, which is taken up from the extracellular milieu, cannot leave the lysosomes. So at first, when we found that, we thought maybe these cells are resistant for viral infection, generally because cholesterol influences fluidity of the membrane, and maybe it's very important also for other viruses. That's not what we found. We found that MPC1, the cholesterol transport function had nothing to do with its role as a host factor, and through work of us and others, we have demonstrated that this is actually a receptor of Ebola virus, the receptor of Ebola virus. So Ebola virus first gets taken up from the extracellular milieu by attachment factors that bring it into the lysosomes. There, cathepsin cleavage occurs of the glycoprotein of Ebola virus, and this exposes a binding domain in the glycoprotein. And before our study, it was known that this binding domain is a receptor binding domain, and we discovered that it binds MPC1, which is abundant in the lysosome, and it transfers, it crosses the membrane, RNA gets released in the cytosol, and then in the case of Ebola virus, all hell breaks loose. So we discovered this new kind of intracellular receptor. After we have done the study, other people have discovered that MPC1, the receptor, plays a big role in adapting the virus to its human host. So it was noticed that in the outbreak of 2014, certain strains of Ebola virus became predominant. And these strains that became predominant, if you sequence them, they got mutations in the viral glycoprotein, very specific mutations. And when these mutations were mapped, these mutations were exactly on the binding site of MPC1 with the glycoprotein. And it was shown that this is a way of the virus of adapting from bat MPC1 to human MPC1. So viruses are important for determining these host barriers. Today I want to focus on a new story where we got intrigued around 2014 with this rare cluster of paralytic disease. So in the outbreak of 2014, there were children that developed these acute flaccid myelitis, and it was a mystery what the causative agent was. It turns out that this occurs in every 2 years, and right now we're in a new cycle of this paralytic illness. And the main culprit of this disease has been demonstrated to be enteroviruses. So enteroviruses are part of the Picornaviridae family. Poliovirus is the most known member of that, and it turns out that these paralysis symptoms are caused by enterovirus 71 or enterovirus 68. Another member of the enteroviruses is the common cold virus, which is the most widespread virus known in humans. And there are no approved antivirals for these for this class of viruses. So what we wanted to do is understand how does the virus infect these cells and what are the host factors that are important for this virus, and maybe then we can develop an antiviral therapy. So we repeated the screen, very similar to the Ebola virus screen. This time we use CRISPR instead of haploid knockout. And we decided to compare different enteroviruses amongst each other. So we did multiple screens with multiple different enteroviruses. We started with a screen for rhinovirus C15. And as you can see, the results are completely different from the Ebola virus screen, and we found some things that are important probably for entry in the heparin sulfate biosynthesis. But this study really wanted to sort of look at things that are important for all enteroviruses. So we repeated the screen with enterovirus D68. There were some differences between these screens. And when you then compare the screens with each other, where on one axis you put the results for the enterovirus screen and on the other the rhinovirus screen, there were basically 3 hits that really popped out. This gene has been known to be important for translation of different enteroviruses. This is a phospholipase that we recently reported on in Nature to be important for different enteroviruses. But we were really intrigued by this top hit SETD3. What is SETD3? It's a histone lysin methyltransferase. So it has a domain that's important for methyltransferase activity and a substrate binding domain. It's not a typical histone lysine methyltransferase, so if you cluster them together, SETD3 clusters in a group that is called the non-histone methyltransferase sets, and it's not known for this particular methyltransferase what the substrate is. In yeast, these are methylating ribosomal subunits. So first we wanted to see how strong is the effect of knocking out SETD3. So we generated knockouts in SETD3. We complemented these knockout cells with cDNA. And as you can see here, the replication of rhinovirus and enterovirus goes from 100% to 0.01%. So it's a real strong effect, which can be reversed by overexpression of SETD3. And it seemed to be important for multiple types of rhinovirus. So rhinovirus can be divided in A, B, and C types, and it's important for all these types, as well as different outbreak strains of enterovirus D68 in 2014. Then we wanted to see in the enteroviruses, how far does this protection, if you knock out SETD3, how far does it go? So we have cells where we knock out SETD3, and this is a crystal violet staining where live cells are stained blue, and these cells are then exposed by different cytolytic viruses. So you see that these rhinoviruses and enterovirus D68 kill the cells. The knockout cells are completely protected, and the add-back kills the cells and makes the cells sensitive again. We saw that also Coxsackie viruses are affected by knockout of SETD3, and Coxsackie viruses are also associated with this non-polio acute flaccid paralysis. We found that poliovirus itself requires this methyltransferase, but not viruses from other families. VSV, chikungunya, and Zika virus are unaffected by this methyltransferase. So it really seems to be an important factor for many enteroviruses, but it doesn't go beyond that family of viruses. If you look at more physiologically relevant cell types, so non-cancer cell lines, human bronchial epithelial cells, especially for rhinovirus and enterovirus D68, those are really the primary cells that are infected. And you can see that knockout really protects these cells quite well for infection. So virus infection is a multi-step process: entry, translation, replication, packaging, and egress. And we wanted to know a little bit more at which step does this methyltransferase act. And for that we use a trick. It's a replicon. These viruses are positive-stranded RNA virus, so the RNA gets translated and replicated immediately in the cell. So you can make it in vitro and just transfect in the RNA, and basically you bypass viral entry. So if you bypass viral entry in the knockout cells, we still saw that there was this dramatic decrease in viral replication. So that led us to conclude that the methyltransferase is not important for entry, so it's not a receptor. It doesn't involve the receptor step, which is usually quite rate limiting, but it's rate limiting for viral translation or replication. And a control where you just have an mRNA that expresses luciferase is unaffected by knockout. So it's really something that is very specific for this enterovirus replication. So to understand a little bit more on the mechanism or which viral proteins are important for this effect, we collaborated with Nevin Krogan and Raul Andino. And in a separate study, they had expressed each individual viral protein and use proteomics to pull down the host proteome that is attaching to the viral proteins. So it's a nice complementary approach. which is also genome-wide. And in their unbiased screen, when they put a specificity score at 0.75, they found 170 host proteins that are important for, or that bind to the viral proteins. We overlaid that with our genetic screen, and we were quite pleased that SETD3 was amongst those interactors. So now we knew that SETD3 binds to this viral protein, viral replication protein. We looked a little bit further in the process of replication and looked at the localization of SETD3. It's in the cytosol and in the nucleus. What we wanted to know is whether the host protein is important in the cytosol or the nucleus, and we did that by directing it either out of the nucleus or into the nucleus with a dominant nuclear export or nuclear localization signal. And we found that you can only restore the infection if you have this host protein expressed in the cytosol. So this goes a little bit against the role or the significance of this methyltransferase as a histone lysin methyltransferase, which is in the cytosol, and suggests that it has a cytosolic function in replication of the virus. Finally, we were interested, is this gene also important for in vivo pathogenesis? And for that, we use a mouse model for infection. And in this mouse model, in neonates, you inject the enterovirus in the brain. It will spread and it will cause paralysis of the hind limb of the mouse. And interestingly, this methyltransferase, a complete knockout of this methyltransferase did not, these mice are viable, so they don't have an apparent phenotype. But if you look at viral infection, normal wild-type mice die, 70% of the mice die if you in this infection model, and the SETD3 knockout mice are completely protected. And on the cellular level, if you look at the viral infection, you see a large decrease in viral infection in the muscle if you knock out this host methyltransferase. So SETD3 is a broad host factor for all tested human enteroviruses in human cells and in vivo. It's important for viral replication, and the localization in the cytosol is required. And SETD3, because complete knockout of SETD3 did not lead to a reduced viability of these mice, you can think that if you transiently inhibit SETD3, you can protect humans against viral infection without big side effects. And with that, I would like to thank the people in the lab, and this was a collaboration with Or Gozani, Nevin Krogan, and Raul Andino. And with that, I will take any questions.
Great talk. Is the methyltransferase activity important for the function of SETD3?
Yeah, so that was the first experiment that we did together with Or Gozani. It's not as easy as with kinases, where you completely can knock out the activity of this methyltransferase. We have done a knockout where we reduced the enzymatic activity with 90%, and that did not affect the viral replication. We cannot exclude that this remaining 10% activity is important, is sufficient for its role. So now we're making more mutations and try to kill the enzymatic activity more strongly.
Great talk. Maybe I missed it. How does the virus hijack the cellular machinery of this very fascinating epigenetic enzyme how does it work? Like, I still can't wrap around.
Yeah, so we know that it tightly binds to the 2A protein, and the 2A protein on itself is required for replication. What we think is that the binding regulates the activity of 2A, and 2A has a zinc binding domain, and we actually think that the SETD3 sort of shields that binding domain, and now we have to find out how that shielding of the domain, how that is favorable for viral infection. So we think it does so by physical interaction. Okay.